Good afternoon and welcome to the Galien's Foundation Week of Biomedical Innovation webinars, celebrating the best of the best in scientific innovation and in the improvement of the human condition. My name is Desmond Murphy. I'm the former head of Global Medical Affairs at Oncology for Servier. I've recently taken up a new position in the US, working across the late development and commercialization of our oncology assets. And I'll be serving as your chair and your moderator this morning, highlighting the discussion around the nominees for the best pharmaceutical product category of the annual Prix Galien USA Awards program. Now, these webinars are taking place as an essential background to the upcoming awards from the Galien Foundation, which will be celebrated in New York City tomorrow evening at the Museum of Natural History. And we're expecting over 900 participants at this event, ranging from biopharmaceutical companies, scientific experts in public health, all there to celebrate innovation in pharmaceutical and biotechnology advancements. We have also the background of the week where the Galien Foundation are running sessions in New York today, bringing together these experts to deep dive into some of the innovation that has come our way over the last year. And it's incredible to see what we've been able to develop jointly as a biopharmaceutical industry, bringing innovation to patients. So before I introduce our panelists, I'm just going to give you a breakdown of the products that are up for this award. We have 18 products overall that are nominated, and ranging from a wide variety of disease areas. So in order of alphabetical order by organization, we have Elahere from AbbVie, which is for ovarian and peritoneal cancer. We have Zefosa from Ardelyx for chronic kidney disease. Zurzuvae from Biogen Sage Therapeutics for postpartum depression. Cobenfy from BMS for schizophrenia. Leqembi from Eisai Biogen for early Alzheimer's. Kisunla from Eli Lilly, again for early Alzheimer's. Vabysmo from Genentech Roche for macular degeneration and edema. Phesgo from Genentech Roche for HER2-positive breast cancer. We've Yeztugo from Gilead for HIV. We've Trivio from Idorsia Pharmaceuticals for hypertension, and we'll be joined by one of their panelists today. We have Obzura from Incyte for atopic dermatitis, and we've Zevtera and Zacduro from Innoviva Specialty Pharmaceuticals, which are antibiotics, and again, we're joined by one of their panelists today. We've Winrevair from Merck & Co. for pulmonary arterial hypertension, Pluvicto from Novartis for prostate cancer, Zabzapret from Pfizer for migraine, and again, we're joined by Novartis and Pfizer panelists today. We've Voquenza from Phathom for heartburn associated with gastroesophageal reflux disease, and we're joined by their panelists. And finally, we have Tzield from Sanofi Aventis for type 1 diabetes. So you see in this best pharmaceutical product category, just a wide range of innovation that we've been able to bring to patients, all up for the award of best pharmaceutical products. So without further ado, I want to introduce our 5 panelists today who are going to talk you through what their products bring to patients in terms of innovation and perhaps some of the challenges that they've had in developing and bringing the products to market. And I'd like to start with Eckhart from Phathom to give a brief introduction. Eckhart, if you don't mind.
Yeah, thanks, Desmond. Let me say first that it's a great honor to be here on behalf of Phathom Pharmaceuticals, and evenly great to be among all these great panelists. My name is Eckhart Leifke. I'm the Chief Medical Officer, and I'm leading medical strategy and drug development for Phathom Pharmaceuticals. We are a relatively young company. We were co-founded by the late famous Dr. Tachi Amada in 2018 and became public 2020. 2019. We brought to the US market Voquezna. The pharmacological name is vonoprazan. It is a novel class of medicine and the first acid suppressant therapy for more than 30 years for erosive as well as non-erosive forms of gastroesophageal diseases, also aggravated GERD, as well as in combination with antibiotics for eradication of H. pylori infection. Voquezna provides a faster onset of action, a more potent gastric acid suppressant, and has a longer duration than
Thank you.
APIs and current standard of care.
Yeah, thank you very much for that introduction. We're very happy to have you as part of the panel. Rod from Pfizer, if you don't mind.
Yes, hi. Good morning, good afternoon, everybody. My name is Rod Junor. I work at Pfizer. I'm the development head for internal medicine. And as part of that role, I lead the clinical development of Pfizer's CGRP portfolio, which are drugs for migraine. I'm delighted to be here. I'm very happy to talk to you about Zavpret, but perhaps I'll wait until I'm asked and then deal with your questions as they come. Thanks, Des.
Yeah, perfect, Rod. Martine?
Hello, I am Martine Clausel. I am MD. I am one of the co-founders of Idorsia. And before I was one of the 4 co-founders of Actelion. I am a Chief Scientific Officer and Head of Research in Idorsia. And I will explain a little bit my link to the research which we have done over 35 years to arrive to Tryvio for treatment of difficult to treat hypertension. And it started in Actelion at the very beginning of Actelion with our work in the field of pulmonary arterial hypertension. We had discovered the first endothelin receptor antagonist, and we published in Nature. And we went through approval of Tracleer and Opsumit in pulmonary hypertension. Our first work on hypertension had been in 1989, but it took all that time to arrive to Tryvio.
Super. Thank you, Martine. And Chaitanya? You're on mute, Chaitanya.
Yes. Am I better now?
Yeah, perfect.
Hello, everybody. Sorry for that glitch in the beginning. It's a pleasure to be here and an honor to be here as well. I'm Chaitanya Devki. I'm a senior strategic expert for therapeutic innovation at Novartis. I joined Novartis about a year ago after close to 40 years working on radiopharmaceutical therapies in various academic centers. The last one being Columbia University. And the first one where I was for the majority of my career being Sloan Kettering Cancer Center. And the reason Novartis believes in radiopharmaceutical, or as we call it, radioligand therapies, is because radioligand therapies offer the unique ability to target a particular receptor in a cancer cell, on a cancer cell, with a payload that is capable of selectively destroying that cancer cell and its immediate environment without a significant side effects or toxicity profiles that enable the patient to receive the therapy and then almost immediately or soon thereafter go about their normal work. We've been focused on prostate cancer, but are also now looking at radiopharmaceutical therapies in various other cancers as well. Thank you, Desmond.
Thank you, Chaitanya. And finally, Shruta?
Good morning, everybody. I'm Shruta Rege. I'm Senior Vice President, Head of Regulatory Affairs and Development Operations at Innoviva Specialty Therapeutics, which is really focused on delivering innovative therapies in critical care and infectious diseases. And I'm really honored and thrilled to highlight 2 of our products and the successful innovative journey of 2 of our products, Xacduro and Zevtera. Very pleased to be here.
Super. Thank you, Shruta. I neglected to say for the audience as well, don't hesitate to put your questions in the Q&A option at the bottom of the Zoom call, because we'll take those after a brief round of deep diving into the various products. Shruta, if you don't mind, we'll start with you to hear a little bit more about the antibiotic products that you've developed. I mean, we're interested to understand the unmet need. I mean, and we know how challenging it is to develop novel antibiotics, and how these products represent a breakthrough for the treatment of patients that you're targeting.
Sure, thank you. I can start with Xacduro. So carbapenem-resistant Acinetobacter infections, or CRAB, as they're often called, it is one of the most urgent antimicrobial threats globally, and it has been identified as a public health threat by CDC and WHO. And these infections are really challenging to treat and often affect vulnerable patients. They have mortality rates ranging as high as 40% to 80%. And as you can imagine, antibiotic resistance has continued to rise. Innovation and investment in these new therapeutic options, particularly pathogens such as Acinetobacter, have really lagged behind. And so we recognize that unmet need and saw an opportunity to really develop our first targeted next generation antimicrobial for this gram-negative pathogen, specifically targeting Acinetobacter infections. Xacduro, our product, also known as sulbactam durlobactam, it became the first pathogen-focused antimicrobial that was approved for hospital-acquired bacterial pneumonia and ventilator-associated pneumonia caused by susceptible isolates of Acinetobacter complex. Xacduro consists of sulbactam, which is a legacy agent with known activity against Acinetobacter, and durlobactam, which is a novel beta-lactamase inhibitor. It's really designed to overcome the modern resistant mechanism. And in our global large phase 3 trial, Xacduro significantly outperformed colistin with a significant reduction in mortality rates. We had higher clinical cure rates as well as a microbiological favorable profile and a favorable safety profile when compared to colistin, which is the prior standard of care to treat Acinetobacter infections. And now, our product has been included in the Infectious Diseases Society of America guidelines. And so because of that, adoption is really accelerating. And this is really driven by the compelling data, the guideline adoption, and the urgent clinical need here. And just talking a little bit about Zevtera and the infections that Zevtera covers. Zevtera is approved for 3 different indications. Today, we're focusing on Staphylococcus aureus bacteremia, which it's really one of the most challenging infections in hospital medicines. Over 100,000 people in the US develop SAB annually. And despite the available treatments, mortality again remains around 30% within 90 days. Until recently, there were no FDA-approved treatments for this particular indication in over 15 years. And so this was the first and only cephalosporin approved specifically for Staph aureus bacteremia, including those with right-sided infective endocarditis. What is unique and special about Zevtera is it's bactericidal. It rapidly clears the bacteria from the bloodstream, which is also a key marker for improved outcomes. And this is really critical for this high mortality indication. And unlike other cephalosporins, it maintains activities against both gram-positive and gram-negative bacteria with broad spectrum efficacy and also a favorable safety profile.
Yeah, thank you very much, Shruta. And it's a fascinating development program, and we'll come back to it because I think we know how challenging it is to develop novel antibiotics. But I'll save that for the next round of questions. Maybe Chaitanya, if you don't mind, take us to a totally different area. I mean, radioligands are a fascinating, I would say, therapeutic innovation that's we're starting to see come through over the last number of years. And in your background, of course, is very interesting, having only recently joined Novartis. So can you tell us a little bit more about the technology and what it brings to patients with prostate cancer in this case?
Delighted, Desmond. As I said earlier, the advantage or the unique feature of radioligand therapy is that you have a ligand which can be anything from a small molecule to an intact immunoglobulin that is specific or preferential to a cancer receptor, a receptor found on cancer cells. And we were fortunate that we were able to evaluate a cyclic peptide labeled with a beta minus emitting nuclide. So what the nuclide does is it deposits cytotoxic energy within a radius of a couple of millimeters. So that offers exquisite specificity in terms of hitting the cancer cell, hitting surrounding cancer cells, the so-called bystander effect, and not hitting adjacent normal tissue to any significant extent. Pluvicto has had the unique ability to cause significant reductions in all the biochemical parameters of prostate cancer, which is PSA 50, PSA 90, etc., reduce and delay the progression of the disease in patients with metastatic castration-resistant prostate cancer. We decided to start with metastatic castration-resistant prostate cancer because in that particular space in the life cycle of prostate cancer, there is very little that we could afford the patient in terms of efficacious therapies. So it was a window that we could test this product in. There were several challenges associated with the product, not least that you have a radionuclide being made in either a reactor or a cyclotron. You have a cyclic peptide being made in a reactor of another sort. And you have to have the ability to attach them to each other in such a way that they don't fall apart. They don't fall apart before administration to the patient, and they don't fall apart by enzymatic degradation in the patient. So there are several logistic and biologic challenges associated with creation of this radioligand. And in metastatic CRPC, castration-resistant prostate cancer, what we found was that the therapeutic profile was really excellent. And this is published, the agent is approved, so I'm not going to go into too much detail there. But what we also found was that we were able to extend the overall survival of these patients with a very bad, aggressive form of prostate cancer. That then encouraged us to look at earlier forms of prostate cancer. So we then focused on patients who had castration-resistant prostate cancer, but who had not yet received and not been benefited by chemotherapy. So the chemotherapy-naive patient population also had a significant increase in their performance characteristics as well as in their overall survival as compared to standard of care. That then encouraged us to look in the hormone-sensitive prostate cancer population, and we found comparable results. So the side effects profile is really quite innocuous. The only toxicity is a dry mouth, which is irreversible, but not very significant. So it has been a significant, I keep saying significant, it has been a remarkable improvement in the delivery of very directed, specific therapy to cancer cells. Thank you, Desmond.
Yeah, it's an incredible technology, Chaitanya. So thank you for sharing that with us. Martine, you're next on my screen, and hypertension is an area that Servier have worked in over many years. So we know how challenging it is to develop, again, breakthroughs in hypertension. So can you tell us a little bit more about Trivio and what it brings to patients with hypertension today?
Thank you very much. Hypertension is a very frequent disease. By the estimation of the people who worked on global burden of disease, it was considered as a #1 modifiable risk factor for early premature mortality and cardiovascular morbidity. And despite the many drugs which exist, about 50% of people with hypertension don't get a good control of their blood pressure. Even if they are compliant, if they take drugs, sometimes they take up to 6 or 7 different classes of drugs, they remain hypertensive. And yet it is such a bad risk factor for strokes in particular. Strokes remain a huge problem. Myocardial infarction are markedly increased by hypertension, and 50% are not well controlled. We had been working on our side on the hypothesis that endothelin, which had been described in 1988 as the most potent vasoconstrictor known, but is much more than a vasoconstrictor, It's also a very strong co-mitogenic, so it's a co-growth factor. It plays a role in vascular hypertrophy, in cardiac hypertrophy, in fibrosis, in inflammation, and in aldosterone production, sympathetic stimulation. We felt from 1989, which was my first paper on endothelin potential in hypertension, that there was a role of endothelin. And our hypothesis was that if many people were remaining hypertensive despite combination of treatments, it was because none of the existing drugs before was tackling this endothelin system, which was upregulated in hypertension and very likely important. It took us a very long time because in our work on endothelin, since the very beginning of endothelin research, we felt we wanted to have really an ideal drug for treating hypertensive people in combination with other drugs. And we arrived to a molecule which was a once-a-day drug, very potent, without drug-drug interaction. So it could be combined without any interference with renal elimination. So people can be in renal failure and there is no change in pharmacokinetics. And we felt that we had attained, after having put 3 drugs in the field of endothelin antagonists on the market in pulmonary hypertension, scleroderma, and cerebral vasospasm and delayed neurological deficit after subarachnoid hemorrhage, we had now the perfect drug for hypertension. In phase 2, we saw that we were beating lisinopril in terms of blood pressure reduction without any side effect, basically, except plasma volume expansion. So in phase 3, we went out to try to find really a way to study patients who were truly resistant. So we made a standardization of a combination of 3 drugs of different classes, and they were all given X4-HCT with a combination of hydrochlorothiazide, amlodipine, and valsartan. And when these people were remaining after a very good control, including of their good compliance, taking the drug by valsartan measurements, we then, if they were still hypertensive, we randomized them to aprocitentan, Trivio, or placebo. And that was a very, very difficult trial to do. And we have seen that aprocitentan achieved at 4 weeks only. The study has been a 48-week study. And of course, you cannot leave people on placebo for 48 weeks. So there was a single blind on aprocitentan for 32 weeks. Which was something quite particular. There was no placebo to put in perspective any side effects which we would see. We see a 15.4 millimeter of blood pressure of mercury of blood pressure reduction in people who have sometimes been hypertensive on 6 or 7 drugs and were all still hypertensive on 3. On top of these 15 millimeters of mercury decrease with a very good safety, no hyperkalemia, no tachycardia, no headaches, no hypotension. And we have overall of 48 weeks an extremely low discontinuation rate. And this blood pressure reduction is probably very important. These people with truly resistant hypertension have a very poor prognosis, basically a twice higher risk of bad cardiovascular outcome as compared to normal sensitive hypertension, in part because probably endothelin is a very bad molecule. When people have an important role of their endothelin system, the prognosis is worse. And we have the first drug ever to treat endothelin in hypertension.
Oh, super. But thank you, Martine, and congratulations, because again, we know it's been many years since we've been waiting for more innovation in hypertension. I think on a similar vein, Eckhart, if you don't mind, GERD is an area I worked in many, many moons ago. We thought that with the proton pump inhibitors, we'd made a major advancement. So I'm really interested to hear what Fathom has done to take therapeutic innovation a step further.
Yeah, thank you. So GERD as well as H. pylori infection affect millions worldwide and also millions just in the United States alone. And significant needs remain despite the fact that PPIs are widely available, and partly due to the limitation of the pharmacological properties. H. pylori, as you all know, can lead to peptic ulcers and is indeed classified by the WHO as a class 1 carcinogen for gastric cancer. We know that eradication of H. pylori, commonly combination of 1 or 2 or sometimes even 3 antibiotics with the PPI, can reduce this risk significantly. However, antibiotic resistance is on the rise. And we also know that earlier and more potent pH suppression, so higher pH in the gastric environment, makes H. pylori more susceptible to antibiotic effects. GERD is clearly linked to obesity and therefore really on the rise, particularly in the United States. There haven't been any advancement of pharmacotherapy for non-erosive or erosive GERD for almost 3 decades. We know that long-standing GERD can lead to erosions, strictures, or even Barrett's esophagus, which further can turn into a pre-form of cancer, which is costly and cumbersome to treat. Proton pump inhibitors, and I don't want to go into too much technical details, have shortcomings based on the pharmacological limitations. And we see this clinically. I think this might be more important for the audience to hear because there's a lot of cycling of patients through different PPIs, always in the hope that the next PPI will provide better relief or better healing of erosions. We also know that actually off-label use of double dosing or BID dosing of PPIs is quite common. And we also know that there's a substantial relapse of patients with erosions under the treatment of PPIs. So we saw with Voquezna that acts differently, slightly differently on the ATP pump and provides a faster, earlier, more potent and more durable acid control as an opportunity to overcome these shortcomings. And I'm happy to talk about the clinical data maybe in the next round of questions.
Yeah, thank you. Thank you, Eckhard. And last but not least, Rod, if you can talk to us a bit more about Zavzpret and the treatment of migraine, again, an area with a lot of unmet medical need.
Yes, of course. And, pain's an area with a very high disease burden. And Pfizer has decades of experience in pain. We've had drugs like Celebrex and Lyrica and Neurontin. And we have a history with migraine as well with Relpax and more recently with Nurtec. So why so much investment and interest? Well, migraine is an extremely common condition. Around 15% of the adult population have migraine. And it is far more than just a headache. It's actually the second leading cause of disability globally. And therefore, it causes lots of disruption to daily life. And for those who don't know, migraine is a moderate to severe headache, but it's not just that, because these patients also get photophobia. They get nausea and vomiting. They get phone phobia. And all of this is aggravated by routine physical activity. And when you get a migraine, it can last for up to 3 days. So, when you think about severe headache, lasting for 3 days that basically doesn't allow you to do anything because it's aggravated by routine activity, there's a lot of unmet need there. And the real breakthrough in migraine came when we started to understand the role of calcitonin gene-related peptide, CGRP, and that became a validated drug target for migraine. But the question is, how do you hit CGRP? There are lots of different ways of targeting CGRP, and you've got to figure out how you're going to hit it, and then you've got to figure out how do you deliver the drug. And the key issue with migraine is that you get the nausea and vomiting in a sizable proportion of patients, and it's over 50%. With this nausea and vomiting, that means taking oral treatments can be problematic because you may be too nauseous to want to take anything orally, or you may take it orally and then vomit. So for patients who are really affected by nausea with their migraine, an alternative to oral administration really makes a lot of sense clinically. And so what's that alternative look like? Well, one of the best routes of administration, if you're not going to go for oral administration, is intranasal. And the key thing about intranasal is that what you're dealing with is a very highly vascularized, large mucosal surface for absorption. and it avoids first-pass metabolism. So there are some very good technical reasons for wanting to go for intranasal administration. And then in terms of the benefits, I mean, the real benefit is speed of onset. So we see drug effects within 15 minutes. And when you're having a severe headache that's incapacitating you, you really want to get back on your feet because you've got better things to be doing with your time than being affected by a migraine. Having a drug with a very rapid onset of action is actually incredibly helpful. And having a drug that you can use for the proportion of patients that are very nauseous and they don't want to take orals is also very helpful. So that's where we came at it from in terms of there's a lot of unmet need here, and this is actually a really nice way of targeting the disease with a unique route of administration, the intranasal CGRP.
Thank you, Rod. That's fascinating as well. So in terms of our next round of questions, I'll start with Martine. I think we'll mix the two, the second and third questions together. I'm interested in the outcomes that you're measuring in your clinical development, and maybe related to that, I'm guessing some of the challenges that you had either from an internal development point of view or in discussions with the regulators. So Martine, you touched on blood pressure reductions in terms of millimeters of mercury, but I'm just interested in morbidity, mortality as well. How do you go beyond that in development? What kind of challenges did you have from regulators in a world where there's many treatments available already for hypertension?
So we wanted to concentrate on a place where there is really a medical need which remains very high, and which is the people who cannot control their blood pressure, whatever they are taking, and where the risk is much higher. What we ultimately found out is that we had been very open in the inclusion criteria. We wanted just people to have proven, and that's completely unique, to have proven resistance in their treatment. And we found out that it was really allowing people with very big comorbidities to be included in the trial. So we ended up with 20% of people who had overt heart failure. But when we were looking at NT-proBNP or other measures of heart failure, we did not go in much depth, but we realized that we probably had 40% of people with early degree 1 or NIA class 1 or 2 of heart failure. We had excluded 3 and 4. But we had 22% of people with CKD 3 or 4. We had 56% of people with diabetes type 2. The BMI was extremely high. We had elderly, the average age was very high, above 65. We realized that the people were and we also had many women, menopausal women. We had African Americans in a high percentage in the US, because those are probably the groups of people with all these comorbidities were very difficult to treat. So we are touching a very, very high medical need where there was no good treatment, and that's why people were sometimes spending years and losing years in trying combination of drugs or different classes of drugs, and they were not working. So we are fulfilling a really big medical need here by having this efficacy, which was really not a given at the beginning. And that confirms the important player, which is endotheliin, particularly in patients at risk, because there is evidence that in all of these populations, the endothelin is playing a role. It's upregulated in CKD, it's upregulated, there are also activating mutations in, sorry, I have a need to stop something.
It's okay. We can't hear it, whatever it is.
Okay, it's ringing all the time. And we realized that we were really fulfilling a medical need, and that endothelin was actually the link between these people who had a cardiovascular pathology and their difficult to treat hypertension because endothelin was not tackled. So I think we are covering in a unique fashion a lot of the remaining medical need in hypertension. In terms of morbidity mortality, it's one of the diseases where you don't have to show morbidity mortality because hypertension is considered as a validated surrogate marker of outcome. And indeed, in our label, the FDA proposed by themselves to write that blood pressure lowering is known to decrease the risk of bad cardiovascular outcomes, in particular strokes and MI. And in our observations, we are continuing to work at the moment in that direction. But we are seeing particularly in CKD, for example, in CKD, we are decreasing proteinuria by up to 67% on top of valsartan in certain populations. We are having probably an effect on eGFR. So even though we don't have a morbidity mortality outcome, we see that when we compare overall our MACE or incidence of heart failure, we have very, very low rates as compared to comparable populations in the past in SPRINT, in ALL-HART, and in other databases. So it's good if we want, or if a partner wants one day, because we are a small company, to do outcome studies, to go further in the direction of any one of these indications. There is a lot to do.
And in terms of your definition of resistant hypertension, was that an easy discussion with the regulators in clinical development in terms of having an agreed definition of resistance?
Yes, yeah. Yes, I think the word resistance is something which may not be used anymore, and that's what the FDA was telling also, because we are basically solving the resistance. So they completely agreed. The problem is that some people have been always using the word resistance for anything, but we took the strict definition, which is showing that when people are shown to be truly resistant when they are well treated, when they are taking at least 3 classes of different antihypertensive at their optimal dose, and that after exclusion of course, white coat effects, that's evident, but also secondary causes, or certain secondary causes, because we probably have a lot of primary aldosteronism in our study, and that's very interesting because we have very, very good efficacy in people with particularly high aldosterone, and we decrease aldosterone concentrations. We have agreed with the FDA, and the FDA themselves have proposed to us to do a program in phase 3, which was composed of this study, and only this study, where we have 2 parts which are pivotal, the first 4 weeks, placebo-controlled, and a randomized withdrawal at the end of the 32 weeks of single blind. And if we had a success on both, showing significant blood pressure reduction versus placebo during the first 4 weeks, and seeing significant blood pressure difference to the placebo, which would go back up in their blood pressure after discontinuation at the end of 36 weeks of treatment, then it would be approvable, and that's what we got.
Super, thank you, Martine.
Very, very stringent study.
Yeah. Eckhart, I'll jump to you because I think there's some parallels maybe to your disease area as well with proton pump inhibitors, which had a lot of success many years ago. Maybe a perception of a satisfied medical need that wasn't really the case. So I'm interested to see the challenges and outcomes and development that you had at Phathom.
Before I talk to the clinical trials, let me briefly point out the 2 key mechanistic differences of vonoprazan, which is a so-called potassium competitive acid blocker, to proton pump inhibitors. Proton pump inhibitors have many, but mainly 2 key shortcomings. They only block so-called food-activated pumps. That's the reason why they have to be taken prior to food in order to optimize their effect, and they have a relatively short half-life. And indeed, as you're probably aware of, the metabolic pathway through which PPIs are degraded have different genotypes, and indeed the prevalence of so-called rapid and ultra-rapid metabolizers are quite prevalent in the Western world, particularly in the US. It might lead to even short half-life, which might explain why you see so often BID or double dosing. Voquezna doesn't have that. It acts on resting as well as food-activated pumps, has a far longer half-life, and it is metabolized differently. So the genotype of rapid or ultra-rapid metabolizers doesn't affect the Voquezna. Voquezna, as a small molecule, is, in my view, a unique example how an improved pharmacodynamic as well as pharmacokinetic profile translates to better clinical outcomes. Well, Voquezna is already included in US-based guidelines for the treatment of non-erosive and erosive GERD, as well as H. pylori infection. One of the reasons might be that we did in phase 3 indeed an active comparator trial for EE as well as for H. pylori. In the EE trial, we saw faster and more complete healing in the most severe forms of erosive gastroesophageal reflux disease. And in the HP trial, we actually doubled the eradication rate in those with resistant antibiotic-resistant H. pylori infection. For NERD or non-erosive GERD, FDA wants you to do a placebo trial. The reason is pretty simple. We know that functional diseases are on the rise. The same is for functional heartburn. And we also know that a placebo control separates functional heartburn better from an acid-driven heartburn. In our NERD trial, we did not even see, of course, superior heartburn relief as compared to placebo, but also a more durable effect. And we were able to reduce, not to say to prevent nighttime heartburn, which is in non-erosive GERD a big issue. And in those who really improved in terms of nighttime, also saw and reported better sleeps and a better quality of life. Beyond these short-term trial-related outcomes, I think it is reasonable to postulate, or at least to assume, that down the road, these might translate also to better long-term outcomes, in particular as the differences we saw in clinically relevant groups in our trials really translate to very low number needed to treat. So therefore, I think it's reasonable to assume that we actually might see with Voquezna in future, less peptic ulcers, eventually less esophageal strictures, and even a reduced cancer risk. But of course, that's something the future needs to show.
Thank you, Eckhart. And Rod, I'll jump to you. You described some of the challenges. You had a novel target, a novel mode of delivery as well. I mean, what were some of the challenges in clinical development that Pfizer faced into with Zavpret?
Well, the thing with Zavpret is it's not just a drug, it's a drug device combination. And so you've got all of the challenges that you've normally got, plus some additional challenges. So, we need to do things like optimize the selectivity for CGRP receptor. But then there's a lot that needs to be optimized with the device. Things like the droplet size, the plume geometry, because intranasal administration only works if you deliver the drug to the correct part of the nose. And so the formulation needs to be optimized as well to maximize the intranasal absorption. And you need a formulation that has got adequate stability as well. You need a device that is going to work across all of the variations in nasal anatomy. I mean, everybody's nose is different, but you need a device that's going to work with all of them. And you need a device that is easy to use without any priming. And so, fortunately, these are all problems that have solutions. You just need to work through the problem logically and define the droplet size, the plume geometry, work the formulation, work the device, and ultimately we were very happy with what we delivered because we've got a drug that works within 15 minutes. So we see an effect on pain in 15 minutes. We see an effect on returning to function within 30 minutes. And if you recall, migraine is the second leading cause of disability globally, and it does really impact people's abilities to do day-to-day activities. We see effects that persist beyond the initial treatment to 24 hours and to 48 hours. So as a route of administration, as a drug that is effective for patients who won't take or can't take oral medications, Zavzpret is actually a very nice treatment option for them to have available, and it's really easy to use. And this is the thing. I mean, it's just super simple. It's just a case of you stick it in your nostril, you press the plunger, and that's it. And patients do appreciate that. And the feedback that we get from them is that they like the fact it's a neat device. They like the fact it's easy to use. They like the fact it works really quickly, and they like the fact that they can just get back to work or get back to day-to-day activities very quickly with it.
And I'm curious, Rod, in terms of things like return to work, are those outcomes that you're capable of measuring in the clinical development program? Are they requested by various payers around the world?
We measure return to normal function. So that doesn't specifically mean work, but it means your daily activities. But you've got to recall that many of the patients with migraine are working age people, and a lot of them will be getting migraines at work. So for them, the return to normal function can be, actually, I'm able to get back to work, I'm able to go back into a meeting, I'm able to do what I was doing, or I'm able to continue doing the childcare, or able to continue doing the shopping, or whatever. But that is super important. It's the return to normal function, because we need to sort of convert migraine from being a debilitating condition into something that's treatable and manageable, and allows patients to get on with their daily lives.
Yeah, thank you, Rod. I'm going to go to Chaitanya next, because Rod described the challenges of intranasal administration. You were describing 2 nuclear reactors that you had to bring together, Chaitanya, to make the product. So I'm very curious in terms of the clinical development challenges that brings to Novartis and to your previous work.
So yes, thank you. The challenges include getting the nuclide in the form that you want it to. So you want the nuclide with a certain amount of radioactive atoms per total number of atoms. You also want the nuclide in a soluble form that enables it to be conjugated appropriately with the ligand that you're looking at. And you want other features like pH, etc. The other thing with the nuclide is that it's constantly decaying. So after production, before conjugation to the ligand, you need to have as little time elapse as possible, so you lose less of your drug substance, if you will. So that's one challenge. The other challenge is to make sure that after you've synthesized the ligand to the nuclide, you then have a distribution mechanism, a supply chain that enables the completed, which is decaying all the time. So if you have 10 units of radioactivity, in a couple of hours, it may be less than that. And it's constantly in a mono exponential fashion, it's going to decrease. So the supply chain issues were the most challenging in getting the radionuclide conjugated to the ligand, and then getting the radioligand after quality control provided to the patient in a form that was acceptable, both in terms of the amount of radioactivity that you are administering and in terms of the viability of the peptide molecule so that it would continue to target. So that supply chain issue was a tough nut to crack. The other tough nut to crack, and they were, I wouldn't describe them as tough nuts to crack, I would describe them as nuts to crack, because when you got people together, the cooperation between various groups, the breaking down of silos was something really remarkable to watch. That also occurred at the treatment site, because most oncologists don't have the licenses or the knowledge to administer radioactivity. So you had to have a collaboration, if you will, between the treating oncologist and the nuclear medicine physician or the radiation oncologist that had the licenses and the know-how to administer the radioactivity. So you had a collaborative impulse that needed to stretch from the production of the nuclide to the distribution of the nuclide and to the administration of the nuclide. So that was one challenge. Which was, as I said, it wasn't a tough nut, it was just a big nut. The other challenge was to adjust the mindset of the physicians and the patient, in the sense that physicians are accustomed to a therapeutic process that, as oncologists, I should say, have a therapeutic process where the side effects are measurable in a certain period of time. The debility to the patient is also evident in a particular period of time. We did not see that with the radioligand. Patients were able to go back to work the day after getting their treatment. I know there's been a lot of going back to work discussed already. It is a really, really important thing for a patient to say, I don't have cytokine release. I'm not, completely fatigued and out of sorts and able to go to work. So the quality of life impact was also very significant in these patients. So that was heartening to watch. The side effect profile, as I said earlier, was very, very manageable, with the dry mouth being about the only symptom that was of concern to the patients, but in none of them was it grade 3 or greater in the common toxicity criteria scale, or very few of them.
Super. Thank you very much, Chaitanya. And I'll finish up with Shruta. Shruta, I think if you've been working in pharma over the last 20, 30 years, we know the challenge of antimicrobial resistance and maybe the lack of companies involved in this area, the lack of incentives to develop. So I'm curious about how, Viva overcame some of those challenges to bring these two products to market.
Yes, thank you. I'll start with Xacduro because that's a unique story. We were really focused on a pathogen-targeted approach, which is quite different and unique in the antibiotic space. And that itself required very close collaboration with the FDA because these are very difficult to treat infections, especially Acinetobacter. It's very rare. And also these patients are critically ill. And so to really design a trial around that, find the right comparator, it's quite challenging. And in order to meet the rigorous standards for safety and efficacy, it was quite challenging to design such a trial. So we had to really work very closely with the FDA to really determine the right trial size, the right patient population, and also align on the statistical considerations for the global clinical trial that we undertook. One of the biggest challenges, though, when we launched this phase 3 trial was we launched it and then COVID hit. So we were conducting that phase 3 trial right in the middle of the COVID pandemic. And as you can imagine, these are patients who are in the ICU. And so we had limited access to ICU. That really slowed the patient recruitment. These infections are already very rare to find, but that really restricted the patient recruitment. But despite these challenges, what we did was we adapted. We pivoted our clinical strategy all in alignment with the FDA, and we completed the trials very successfully, delivered very strong data showing a reduction in mortality rates compared to the comparator, as well as very high clinical cure rates and a microbiological favorable profile. So we were able to achieve all these things that ultimately led to the FDA approval. Again, what I say is this experience was quite unique, and it really reinforced the importance of flexibility, collaboration, innovation. I think what was special was there was an article that was authored by the FDA where they provided their perspective, and they actually cited the development program for Xacduro as a model for the successful use of a flexible development program for antimicrobial drugs, especially that are aimed at addressing very high unmet medical needs. Then I'll move to Zevtera because that's also quite unique because that was, Zevtera was developed as a result of innovative clinical chemistry with the goal to develop a cephalosporin with specific activity against methicillin-resistant Staph aureus, again, which is resistant to other cephalosporins. So again, conducting that trial is quite challenging. There was no double-blind, randomized, non-inferiority trial that had been conducted in over 15 years, and so again, we worked very closely. Our partner, Basilia, who developed this product, they actually worked very closely with the FDA to incorporate some novel statistical considerations and trial design into the clinical trial. One of the boldest decisions that Basilia did was choosing daptomycin as the comparator, because it's really more it's newer, it's the formidable antibiotic rather than vancomycin. And again, as you can see, the trial yielded data that is really reflective of the current clinical practice in treating resistant and persistent Staph aureus bacteremia. So challenging, but again, what we did and our partner did was highlighting the importance of close collaboration with the FDA, maintaining that scientific rigor, and the importance of just a flexible clinical development path.
Yeah, thank you. No, it's fascinating to see, and I think we've seen a few examples from the panelists of breakthroughs that, in disease areas that have been maybe 15, 20 years since their last major breakthrough in innovation, which gives us, I think, massive encouragement and hope for the future, because we know clinical development is a road full of failures to get these successes, and I just congratulate everybody on bringing these drugs to patients across the world, because that is our biggest challenge, and that's what helps us get out of bed every morning and do what we do. We've come to I think a natural end. We've got a couple of minutes to go, but I'm going to wrap it up there. So I want to first of all thank immensely our panelists. It's again fascinating to see the variety of innovation that is up for nomination for the best pharmaceutical product category, and I wish everybody the best of luck tomorrow evening in New York to bring home the prize. A big thank you as well to the Galien Foundation for giving us the opportunity to dive a little deeper into the products and to understand all of the efforts that have been made to bring these innovations to market. So for those of you who are in New York tomorrow evening, I look forward to meeting you in person, and a big thank you for your participation this morning.
Thank you, Desmond.
Thank
Thank
you.
you.
Good luck
Thank
to
you.
everybody.